Hello all, I wanted to give you a rundown on the objectives for the final exam. And so in the next few minutes I'm going to cover some of the topics that we've discussed and how they relate to these objectives. And then I will talk a little bit about the cumulative section, which you can see kind of hiding in the lower part of this file. So we've really focused on two main areas that have a lot of overlap with each other. We've talked about the immune system and pathogenicity, and we've talked about vaccination, or acquired immunity through artificial means. In total, there are 10 objectives listed here. First, we've talked about the process of phagocytosis and its contributions to immunity. You should be able to describe the individual steps of phagocytosis, how a pathogen is recognized by a macrophage or a neutrophil, and also be able to describe what might happen to those products after the phagocytosis has occurred. For example, neutrophils just remove that material as waste. But macrophages, on the other hand, or professional antigen-presenting cells like macrophages and B cells, will present that pathogen on MHC2, where it can then interact with T helper cells. Next, we talked about inflammation. And so I'm asking you to describe how inflammation contributes to the immune response. We talked about the role of macrophages to recruit neutrophils through the secretion of chemokines, and also to activate neutrophils through the secretion of cytokines. And we talked about terms like margination, diapedesis, edema, as mechanisms by which neutrophils move to the site of infection, make their way out of the blood vessels and into the site of, into the location where the infection is occurring. And don't forget, we also use the term vasodilation here as well. We also talked about how virulence factors have a role in pathogenicity, and we've talked about a number of virulence factors. In the last lecture on immunology, or lesson 4, we talked about the role of pathogenicity factors such as endotoxins and exotoxins. We talked about adhesins and invasins. But there are other ones we've discussed as well. For example, we've talked about the role of streptokinase in helping streptococci from being basically walled off in an infection by the formation of blood clots. Other organisms like Staph aureus produce coagulase, and that coagulase protects them, kind of like a cloaking device, and protects them from phagocytosis by macrophages. And there are other examples of this that we've seen as well. For example, the role of herpes viruses in repressing the expression of MHC1, which avoids then the immune detection of cells that are infected with herpes virus, and it prevents them from being recognized by T cytotoxic cells. Speaking of T cytotoxic, you should be able to explain the immunological function of them and T helper and B cells. Be able to know what their function is and how they perform that function. For example, B cells secrete antibodies, but they don't do so until they've recognized a particular epitope or a particular piece of a pathogen on the surface of their cells. T helper cells regulate the immune response, and T cytotoxic kill cells. You should know how T cytotoxic cells recognize and kill infected cells. And you should be able to compare the role of a T cytotoxic with an NK cell. Remember that NK cells, unlike TC cells, recognize molecular patterns. So NK cells are recognizing patterns and are part of the innate immune response, whereas the TC cells are part of the adaptive immune response. Finally, you should be able to describe the structure of antibodies and how that structure contributes to their function in enhancing phagocytosis. Likewise, you should be able to explain how T helper cells, through the secretion of cytokines, enhance phagocytosis as well. You can see as I was going through this first section here of the immune response and pathogenicity that there's a lot of overlap. In fact, these objectives are quite inclusive of one another and kind of flow into one another. And so I've broken it apart here for you, but you should know that there is interconnectedness between them. We also talked about vaccinations. Though we didn't discuss it directly in class, you should be able to identify a form of immunity as being passive or active, or artificial or natural. Natural acquired immunity are cases in which an individual is directly being exposed to a pathogen through a natural infection or being exposed to antibodies through, for example, mother's milk. Passive immunity is where you're receiving antibodies or the immune response from somebody else, whereas active immunity is where your own body is producing an immune response. You should be able to compare the different types of vaccines and or describe the vaccines and how they stimulate the adaptive immune response. For example, you should be able to know the differences and the similarities between an attenuated and an inactivated virus or vaccine, and you should be able to describe how are they different in their stimulation of the immune response. For example, which of the two stimulates T cytotoxic cells? You should be able to describe the theory of herd immunity and the function of its design, which we covered in our last lecture on vaccinations. And then finally, you should be able to describe or list reasons why people choose not to vaccinate and strategize how to talk to them. What's not shown here are the objectives that are being created by you. Those objectives from the last two classes that we will have that you are creating as part of your mini lectures will be included here as well as fair game for the final exam. And as I've mentioned before, my questions on the final exam that are multiple choice and are fill in the blank are going to be based off of the questions and the mini lectures that you put together. So you should be able to pay attention to those and be able to address the objectives that have been designed by your classmates and yourself. Finally, I want to address what's going to be on the cumulative section and what will not be on the cumulative section. The cumulative section will be a combination of multiple choice, matching or fill in the blank. There may be a couple of true and false questions. And this here is a list of the topics that may be covered. That can include morphology and gram status comparisons. For example, do gram-positive or gram-negative organisms have a thicker peptidoglycan? Which of them has peptidoglycan? And the answer to the first is gram-positive, and the answer to the second is both of them have it. You should be able to describe the metabolism of a microorganism. For example, if an organism grows best at 37 degrees, you should be able to define it as a mesophile. And so I'd like you to go back and relearn or just refresh yourself on the terminology that we've used throughout the semester to describe microorganisms. You should also have a general knowledge of fermentation and its purpose, and also the final electron acceptors and the processes of glycolysis, and also in the electron transport chain. You should be able to also classify metabolism as being fermentative, aerobic respiration, or anaerobic respiration. I'm not, as before, not going to ask you about specific steps of glycolysis, the Krebs cycle, nor am I asking you to go back and memorize the steps of the electron transport chain, but have a general understanding of it. I mentioned already that I'd like you to have an understanding of the growth characteristics. I should also mention about metabolism that I'd like you to have an understanding of basic terms defining enzymes such as active site, substrate, competitive inhibitor, and non-competitive inhibitor. You also should know, in general, how do you control microbial growth? We talked about physical and chemical methods to control growth, and so you should have a basic understanding of that information as well. You should know the basics of the central dogma. For example, which process uses DNA polymerase? Which process uses a ribosome? And with that said, you should also know or understand the basics of DNA base pairing and the interaction between template DNA, mRNA, and template DNA and non-template DNA. You should have a basic understanding of the horizontal gene transfer mechanisms and how they are different or similar from one another. Should be able to basically list a couple of key characteristics of each. You should also be able to identify mutations and describe them as being silent, missense, or nonsense. And then finally, you should have a basic understanding of antibiotics, antibiotic resistance, and measuring the effectiveness of antibiotics. I'm not going to go back and ask you to memorize the targets of the antibiotics that we've talked about, but it is fair game for me to ask you, for example, that if an enzyme, for example, chloroquine, fluoroquinolone targets gyrase, and I say gyrase is a part of DNA replication. Let me take that back. I'll scratch that. I might ask you, if an antibiotic targets gyrase, what process will it inhibit? And you would have the option of translation, transcription, or replication. And of course the answer here would be replication. I might ask you to measure a zone of inhibition. I might ask you to measure the results of a broth dilution assay and determine which antibiotic is more effective. Things that won't be on here are some of the general chemistry concepts that we've covered. I won't be going back and asking questions specifically on the microbiome, nor will I necessarily be asking questions about microbial evolution. But the basic concepts that we've talked about in these sections may be covered on the exam. If you have any questions, please feel free to ask me. With that, have a great afternoon, and I'll talk to you all soon.